We left off talking about the formation of urine in mammalian kidneys. And so, just to review for a moment, this is the cartoon version of the structure of the mammalian nephron. This is situated within the kidney. The outer layer of the kidney is called cortex. The deeper layer is called the medulla. And the nephron consists of a long tube that starts with the renal corpuscle and continues through the kidney tubule, through segments including the proximal tubule, the loop of Henle, the distal tubule, and the collecting duct. And the kidney tubule is surrounded by a network of capillaries, or very small blood vessels, known as the vasa recta. And those are involved in reabsorbing substances back into the bloodstream, and also in a little bit of secretion of substances that are not initially filtered at the glomerulus. So we described how filtration occurs within the renal corpuscle. The capillaries of the glomerulus are very leaky, and there's a hydrostatic pressure gradient wherein the blood pressure is greater than the surrounding pressure in Bowman's space, and that causes water and various small to medium solutes to move out of the capillaries and into Bowman's space. And that causes flow of liquid along the length of the kidney tubule and through the tubule. So it keeps a continuous flow going through the rest of the tubule. And remember, we identified a higher hydrostatic pressure in the capillaries than in Bowman's space as the driving force for this filtration. So that leads us into the proximal convoluted tubule, and we can think about what happens there. And in the proximal convoluted tubule, we have a great deal of reabsorption taking place. So this is driven by what is by now becoming your favorite protein, the sodium potassium ATPase, which pumps sodium out of across the basolateral sides of these cells into the interstitial fluid in exchange for 2 potassium, and that costs an ATP being broken down to ADP. What that does is it creates a low sodium concentration in these cells, and so that sets up a gradient of sodium concentration between the lumen and the cytoplasm of the cells lining the proximal convoluted tubule. That in turn allows sodium to move passively down concentration gradients through sodium channels, and so sodium moves from the lumen into the cells by passive transport through channels, and then gets pumped out actively across the basolateral membrane into the interstitial fluid. Sodium is the most abundant solute in the tubule lumen, or at least the most abundant ion in the tubule lumen. And so if a lot of sodium is being removed from the lumen, what's that going to do to the osmolarity of the fluid in the lumen? Pause the recording for just a moment if you need to think about that, and think about what that will cause in terms of water movement by osmosis. Okay, so the movement of sodium out of the lumen will tend to reduce the osmolarity of the fluid in the lumen, just ever so slightly, and that will create a gradient that tends to drive water movement from the lumen into the proximal convoluted tubule cells, and then subsequently into the interstitial fluid. And we should note that water cannot readily cross cell membranes, or at least it can't readily cross the lipid bilayer itself. It needs to move through water channels called aquaporins, because water is a polar molecule. So there are abundant aquaporins in the cell membrane that allow water to move down its osmotic gradient, or rather move from an area of low osmolarity to an area of high osmolarity. And so it's following its gradient passively into the interstitial fluid. Now, if a lot of water is leaving the tubule lumen and being transported into the interstitial fluid, what will that do to the concentration of other ions, such as, say, chloride, calcium, potassium? What about all of those other ions? So basically, if we're reducing the volume of fluid in the tubule, we're effectively concentrating all of those other ions. And so that creates concentration gradients that tend to move those other ions passively out of the lumen as well. So chloride in particular moves through chloride channels. And then various other ions such as potassium and calcium also follow along and leave the tubule lumen and get transported into the interstitial fluid. So the overall effect of all of this is that there's a substantial reduction in the volume of fluid in the tubule lumen as it moves through the proximal convoluted tubule, and very little change in the osmolarity. I mentioned a drop in osmolarity due to sodium transport, but it's a very small drop, and because it then leads to a lot of movement of water, essentially the osmolarity that starts at about 300 milliosmolar where filtration occurs in Bowman's capsule, it's still about 300 milliosmolar when it flows out the other end of the proximal convoluted tubule. There are some other important substances that are reabsorbed in the proximal tubule, and those include glucose and amino acids. Glucose is, of course, energetically valuable, can be broken down to produce ATP. And glucose can be transported by a cotransporter that transports sodium down its concentration gradient in exchange for moving glucose against its concentration gradient. And so this is a case of secondary active transport. Glucose is moving up a concentration gradient, but it's being carried along with sodium moving down its concentration gradient. And this is called the sodium-linked glucose transporter, or SGLT. I don't know why it's not SLGT. Once the glucose is in the cell, it still needs to get out the basolateral membrane, and it does that through another transporter, which is called the GLUT transporter. And this was secondary active transport here because glucose was moving up a concentration gradient from low concentration to relatively high concentration in the cytoplasm. But then moving out across the basolateral membrane, it's facilitated diffusion. Glucose is moving down a concentration gradient. It just needs some help to get across that lipid bilayer because glucose is a polar molecule. So it needs a carrier protein to help move it across the membrane. So that's facilitated diffusion. And this is the pathway by which glucose is transported out of the tubule lumen. And it's very similar to the mechanism for transport of amino acids. Those are moved into cells by cotransport with sodium or protons. Okay, so basically over the length of the proximal convoluted tubule, about 2/3 of the urine volume is reabsorbed. And I should mention that the volume that's filtered at the glomerulus is really enormous. So it's on the order of 180 liters per day for a human. If that number doesn't sound impressive because you can't quite picture a liter, basically a liter is about a kilogram if we're talking about water, and a kilogram's 2.2 pounds. So we're talking about 400 pounds of fluid per day that get filtered. If we peed out all 400 pounds of fluid, there wouldn't be much left of us at the end of the day. So obviously we need to reabsorb the vast majority of that. And so in the proximal convoluted tubule, that's reduced to about 1/3 of the original filtered value. But we still haven't really altered the osmolarity of the urine or done anything to concentrate it overall. And if we want to get rid of an adequate quantity of nitrogenous wastes, we need to be able to produce concentrated urine. So how do we do that? And that happens along the rest of the pathway. So we can just have a cartoon version of the nephron here. That includes the Bowman's capsule over here, where filtration occurred. And it includes the proximal convoluted tubule, the distal convoluted tubule, and a portion called the loop of Henle. And the loop of Henle extends down into the medulla from the cortex where it begins. So instead of starting with the so-called descending limb of the loop of Henle, I'm actually going to skip ahead and talk about what happens in the ascending limb, where fluid is moving back from the medulla up toward the cortex. Because in this section, there is active transport of solutes. So active transport of sodium against a concentration gradient, and then that's followed by transport of chloride. And so you can think about what that active transport does to the concentration of water, or the osmolarity of the fluid in the tubule, and then what gradients that would set up for osmosis. Pause the recording for just a moment if you need to. Okay, so if you say that the osmolarity in the tubule goes down, and that creates a gradient favoring water efflux, or water moving out of the tubule, that's correct. But in fact, the membranes of the cells lining this portion of the tubule are impermeable to water. And that's shown in this cartoon diagram by the thick yellow lines. So there's a gradient that would tend to favor water moving out of the tubule, but it can't get out. And so as a result, as all of those ions are pumped out and the fluid continues to move along that pathway, it gradually gets more dilute as it moves along the pathway. And so when it gets into the distal convoluted tubule, its osmolarity has dropped to something in the ballpark of 100 milliosmolar. And remember, that's compared to about 300 at the point where it's filtered, or at the point where it moves from the proximal convoluted tubule into the loop of Henle. We can also think about what all of this added solute is doing to the interstitial fluid along the length of the loop of Henle. And all of that extra solute is gradually increasing the osmolarity of that interstitial fluid. So osmolarity is going up here. And in fact, and this is happening all along the length of that pathway. So we've got sodium coming out along here as well, and chloride. And so it sets up a gradient gradually that causes the medulla to have a much higher osmolarity than the cortex. And so that can be in the ballpark of 1,000 or more milliosmolar deep down in the medulla. And so there's a gradient of osmolarity from the medulla up to the cortex. So now we can turn our attention back to the descending limb of the loop of Henle and think about what happens to the filtrate in the descending limb of the loop of Henle as it flows along and enters an environment where the surrounding interstitial fluid has a very high osmolarity. What kind of gradient would that set up that might drive the movement of water, either in or out of the tubule? Okay, so if you noted that the osmolarity inside the tubule is lower than that of the surrounding interstitial fluid, that's correct. And that would tend to drive water movement out of the tubule. So there'd be net diffusion of water, or net osmosis, out of the tubule. So we've got water leaving the tubule all along this pathway. And so that tends to concentrate the filtrate as it moves along. And so it approaches basically the same osmolarity of the surrounding interstitial fluid as it gets to the base, or the end of the loop of Henle deep down in the medulla. So we've got this very concentrated filtrate down in the medulla, more dilute filtrate leaving the loop of Henle and entering the distal convoluted tubule. So what happens when that filtrate then reaches the collecting duct? And an important point that is maybe hard to recognize in this cartoon version is that all of these tubules are kind of packed in next to each other. So if you think about a box of soda straws in different colors, all sandwiched in next to each other, where the bright red ones represent the ascending limbs of the loop of Henle, and green soda straws represent the collecting ducts, and blue ones represent descending limbs of the loop of Henle. They're all kind of mishmashed in there together. You might think about a box of spaghetti noodles. So the same high osmolarity that we drew in over here, adjacent to the loop of Henle, also applies to the environment surrounding the collecting duct. So as our dilute fluid enters the collecting duct, which way will water tend to move across those cell membranes? So the osmolarity inside the tubule is lower than the osmolarity outside the tubule, and so that would tend to favor water movement out of the loop of Henle, out of the collecting duct into the interstitial fluid. And so all the way along this collecting duct, water is leaving, and that movement of water, along with a little bit of additional solute transport out, greatly reduces the volume of urine that is, or the volume of filtrate or urine, whatever you want to call it at this point, that is moving down the collecting duct, and concentrates it. So depending on how much it gets concentrated, it can reach very high osmolarity by the time it leaves the collecting duct, greater than 1,000 milliosmolar. And so a very concentrated urine moves into the ureter and ends up getting stored in the bladder to be excreted later when it's convenient. So we've now described a mechanism that can produce a very concentrated urine and relatively small volumes of that urine. And we've really primarily talked about the movement of sodium and chloride and water, but one of the main reasons that we need to urinate is to get rid of nitrogenous wastes. And so there are also fairly high concentrations of urea contributing to the osmolarity of the urine, particularly when we're producing a very concentrated urine like this. Now, we don't always produce very concentrated urine. This is under the influence of hormones, and we can fairly quickly switch between producing a concentrated urine if we're dehydrated and producing a more dilute urine if we take in a large quantity of water. And so how does that happen? This depends on the actions of multiple hormones. We'll just talk about one of those for the moment, and that's a hormone called antidiuretic hormone, or ADH. So we drew in a collecting duct where we had dilute fluid coming in, experiencing an osmotic gradient such that we tended to have water movement out across cell membranes, or I should say across a layer of thin epithelial cells that line the collecting duct. And in order for water to move across those lipid bilayers of those cells, what did we say was required? So water is a polar molecule, can't readily cross the lipid bilayer itself, and so it had to move through water channels that are called aquaporins. And so in the presence of high levels of antidiuretic hormone, there are plenty of aquaporins. And those are, specifically we can say there are plenty of aquaporins in the apical membranes of these cells. And the apical membranes are the membranes that are aligned toward the lumen of the tubule, as opposed to the basolateral membranes oriented toward the interstitial fluid. On the other hand, when levels of ADH are low, then that puts the animal in a state called diuresis, or there's a diuretic effect occurring. And so now we have this fairly dilute fluid coming in. We've got the same gradient here, but these cells have very low levels of aquaporins, or almost no aquaporins. Very few aquaporins in their apical membranes. And so even though there's a gradient that would tend to favor water movement out of the tubule, a difference in osmolarities, there's just no pathway for water to move out, and so the water stays in the lumen. And if the water stays in, then the flow rate increases, large quantities of urine are produced, and the osmolarity of the urine is relatively low, so less than 100 milliosmolar. And there's also a little bit of active transport of solutes out of the collecting duct that can even reduce the osmolarity a little bit below what it's at when it leaves the distal convoluted tubule. So this switch can happen fairly quickly. If you drink a couple of quarts of water all at once, then you'll notice within, definitely within an hour, you'll probably find yourself peeing more than you were before. And on the other hand, if you sweat a lot, if you go and play an hour of basketball or go out for a run on a hot day, you'll notice that you produce less urine fairly quickly. There's some other effects going on during exercise as well. So let's say you're sitting in a sauna. We could take the exercise out of it and just have you sweating for some other reason. But yeah, any dehydration will tend to cause you to reduce urine production. So we can think about putting this in the context of a regulatory feedback loop and thinking about what each of the components of the regulatory feedback loop would be if it's regulating our plasma osmolarity and body water content. So I'm not going to go through all those steps. We talked about feedback loops governing some other processes and some other aspects of the system. So I'll let you work on this on your own. Think about what each of those components might be. And as before, there might be some elements that you can't really pin down with your current level of experience with physiology, but you can be a little bit vague about those. Another kind of core principle I wanted to talk about is the idea that homeostasis involves dynamic exchange. We mentioned this in the context of temperature regulation. It's true for water balance as well. Even though we might keep a consistent body water content from day to day and week to week, we probably each take in a couple of liters of water every day, either in the form of liquid water or water in our food, etc. And likewise, each day we excrete a couple of liters of water in the form of urine or sweat or respiratory water loss or feces. Actually, all of those components contribute to water losses. So we're not keeping the same water molecules week after week, month after month, year after year, but we are keeping approximately the same amount of water by replacing what is lost. All right, and then just for fun, I added in 2 more slides on how the duration of urination scales with body size in mammals. But before we look at some data, just take a guess at this. How does the time it takes to urinate vary as a function of body mass in mammals? So pause the recording for just a minute and sketch something in, and then we can look at some data and see if your instincts are right. Okay, so hopefully you've got a good guess recorded. Let's take a look at the data. These data are from a paper that was published in 2013. And do they match up with what you predicted? So if we focus on the mammals larger than about a kilogram, about 2 pounds as our lower cutoff, we can see that for the most part, urination duration is independent of body size. So larger animals pee for about the same amount of time as smaller mammals. Very small mammals are sort of in a different category. When you get down to the size of a rat or a mouse or a bat, they don't pee as long. But once you get over about a kilo, and mammals pee for something on the order of 20 to 30 seconds. It's not completely independent of body mass, but it's pretty close. And it sort of makes sense when you think about it. So if you think about a 70 kilogram human peeing for 20 or 30 seconds, that's about 1/3 to 1/2 of a second per kilogram. Now what if we think about a 9,000 kilogram elephant doing the same thing? If it peed for 1/2 a second or 1/3 of a second per kilogram, that'd be something on the order of 3,000 to 4,000 seconds. How many seconds in a minute? 60. How many minutes in an hour? 60. So 3,600 seconds in an hour. If an elephant peed for the same number of seconds per kilogram that we did, it would take it an hour to pee. Basically, by the time it finished peeing, it would need to pee all over again. And yeah, it would just kind of walk around dribbling all over the savannah. So it looks like basically mammals all pee for about the same amount of time. And this made some newspaper headlines when it came out in 2013, 2014, basically saying that all mammals pee for 21 seconds. If we look at the scaling exponent, it is slightly greater than 0, so it's not quite true. And of course, for any allometric relationship like this, there is some variation. Not to mention that these researchers made observations at the zoo. I don't know how much data they collected for each species, but obviously there's some individual variation. You don't pee for the same amount of time every time you go to the bathroom. So some individual variation, but overall not a strong change as a function of body mass. So there's your fun fact for this week. We'll stop there and move on to some other topics.